Welcome, everybody. Ben is traveling. I'm your interim host, but I will pass it off very quickly to Marcelo. Marcelo is a behavioral ecologist, um, working on understanding evolutionary and cultural drivers of behavior, focused on animal communication systems. I think probably lots of people here are in that world. Um, His research combines behavioral studies, comparative phylogenetic methods, and advanced data analysis techniques. So we're going to talk about an R package today that he has designed to streamline the analysis of habitat-induced degradation in animal sounds. So super psyched. These always do really well. We've got a lot of attendance. Marcelo, I think this is your second or third BioAcousTalk, talk, maybe. Um, I know you've done it before, but very glad to have you and take it away.
Well, thanks, Cameron. And yeah, thank you for inviting me. Yeah, this is the second time. I was talking about our packages. Yeah, so I'm gonna be talking about baRulho, which is a Portuguese word, means noise. And this is a package that aims to help users to measure the degradation that the sound suffer when traveling when propagating in general, but for us, for biologists, we want to understand that in natural environments. Um, and first, I want to give you some sense of why we want to use such package. And this is related to, I guess, the research I do. There are many other things that can be done, but I'm pretty biased. So I'm going to talk about how signals degrade, and then how we think that has something to do with the evolution of the signals, right? So we know this when sound is propagating in space, it will degrade for sure, just for spherical spreading. But also when you have lots of obstacles in the environment, you have things like reflection, absorption, scattering, just to mention a few interactions with those obstacles that are going to affect the structure of the signals as they will be perceived by the receivers, right? Um, so those are additional sources of degradation related to those obstacles that we see in the environment that then we can expect to affect the communication value of the signals. And we know that natural habitats are full of obstacles, right? So the vegetation structure can be quite dense in different environments, and also it could be very diverse. Like different environments are gonna have really different arrays of vegetation, which we can think as being like unique acoustic selective regimes, right? Like they are shaping, or at least exerting the selection force on the signals that are being transmitted in those habitats. And we expect that to have some weight in the way that the vocalizations and the sounds that are produced by animals living in those particular environment have evolved. Their actual expression, the current expression, I should say, of those vocalizations. All right, so we can think about these signals being degraded in different ways, different habitats, and then we expect that to work differently and to those different habitats serve like specific selective regimes on those signals, right? And then we expect those signals, at least during evolutionary time, or it could be during cultural evolution, to respond to those selective pressures in a way that they will be finely tuned to those acoustic environments, to maximize their effectiveness. In many cases, effectiveness is about propagating as far as possible and reaching the audience in a way that um, they can get the information that is encoded in the vocalizations, right? So this is from an evolutionary perspective how we think is important sound degradation for understanding signal evolution in natural environments. All right. And something cool about this is that we can actually test these ideas in an experimental way, right? Which is not always the case with behaviors, right? Like we're talking about behaviors here. I know like most people come from passive acoustic monitoring. I'm sorry, this is not exactly about that, but it's other things that we can do with sound, right? To understand where those sounds come from in terms of the evolutionary process that created them, generated. All right, so for these specific behaviors, we are lucky enough that we can do experiments. Once we register them, recorded them, then we can play back those sounds in specific habitat and then re-record it, right? So this is like experimental design that is common to understand how these sounds degrade in particular habitats. And with that, we can quantify the amount of change that the signals suffer by uh, while propagating in those habitats, right? By comparing what was played against what was re-recorded as increasing distances, we can have some idea of how they changed, which features were affected, in which way they were modified, and so on, right? So this is a pretty cool thing that we can do with sound. But it's also not that straightforward. It's a tricky thing to do because it involves several steps, right? You need to format and manipulate the files. Ideally, you want to take all the sounds you want to test and put them in a single sound file with similar intensity of different signals. Then you need to go out and broadcast those signals and re-recorded them in a standardized way in the natural environment, which is also not straightforward. And once you have that, you need to take those recordings and annotate them, right? Find the signals, the re-recorded signals, the recorded sounds. And it can be tricky, mostly because we are expecting them to get degraded, right? So it's not that easy to find things that can get blurry. It's hard to see in the spectrograms and so on. And once you do that, you can quantify the degradation, right? That's usually the end goal of this type of experiments, to add some numbers that tells you which ones get more degraded and which aspects of the signals were more degraded and so on. So it's a very particular experimental design that I think these difficulties for implementing them have contributed to this type of experiments not being that common in bioacoustics. So people haven't used it that much, and I feel like it's in part because it gets tricky to do, right? Um, so in terms of computational tools that allow people to do this or help people with this, there is not much, and I'm just gonna talk about for which steps of this process, and this diagram that we have here is basically the same things that I just talked about, like different steps that are involved when you do this type of sound degradation experiment. From obtaining the sounds, can be recording or synthesizing sound, to put them in a sound file, master sound file, broadcasting outside, recording, annotating, and then quantifying, right? So these steps, there is only one, well, as far as I know, and I think have a good sense of this now. There was only one piece of software that helped people with this type of data analysis and manipulation, which was SIGPro. It's like software from the early 2000 or around that time. It's been around for a while. It's a graphical user interface software that allows people mostly to annotate the re-recorded sounds manually, one by one, and then do some quantification of the degradation. Right, so that was the software that, to my knowledge, we had available as a community for doing this kind of experiments. So I'm gonna be talking about this our package, new package, the baRulho package, which offers some additional tools well, to help to make life a bit easier for people running this type of analysis, from creating more synthetic sounds to putting those sounds together in master sound files, aligning or annotating the sounds, making sure you are getting the right sounds from those degraded signals, and adding graphically to double check you are doing annotating the right sounds, and then to quantify the degradation with several metrics, 8 different metrics, I think, or a few more, that will allow you to well, finally get some numbers about the amount of degradation you have in those signals. All right. And for this, so the goals of this talk are gonna be, of course, I'm gonna showcase the package, but I'm gonna do that while talking about specific experiment we did on looking at the effects of habitat and signal structure on the transmission of synthetic sounds, right? So I'm gonna go through the different tools that we have in the package, but at the same time, I'm gonna work or show you the results for this particular experiment. Then I'm gonna compare some of the well, the results that we have with baRulho to those from SIGPRO to have some idea of how much we are kind of in the same track, getting similar measurements, and also the performance of the two software compared. And at the end, I'm just going to talk about some additional examples of research using baRulho. Actually, one, because we don't have much time, but I'm going to talk about another example of research on baRulho. All right, so the first step, as I said, in this case, at least for this example study that we're gonna talk about, we created synthetic sounds that kind of mimic or look like, resemble the shape of the structure of animal sounds. And we can do this with the function synth sounds that allows you to vary, to modify specific features of the signals. For instance, here we're creating well, 3 sounds, each one with a different frequency. So the argument here, frequencies, I'm not gonna be talking about specific code. So frequencies is in kilohertz, and this is the spectrogram of the object, the sound object that is created in R. So I'm creating a synthetic sound with 3 frequencies, 1, 2, and 3 kilohertz. You can add more and more varying features to this. For instance, you can have durations, you can have the signals having different durations. In this case, is 0.1 and 0.2 seconds. So notice that we have now the short ones, 0.1 seconds with the 3 frequencies, and the long ones, the 0.2 seconds, also with the 3 frequencies. So the function will produce synthetic sounds for all possible combinations of features that you want to add variation. You can also add frequency modulation, and there are different ways in which you can tune this to, like to change the amount of modulation you get. But like frequency modulation is about how much change in frequency to time you have in a signal. So we have the same 6 signals here without modulation, and then another 6 signals at 1 to 3 kilohertz, 0.1 and 0.2 duration, but in this case modulated. And you can also add amplitude modulation. Amplitude modulation is more about the change in the intensity of the signals, right, through time. So you can actually see the amplitude modulated signals here at the end of the sound. So basically the same sounds that we have over here, but in this case, you can see in the amplitude envelope how they go up and down. So basically having 2 bumps on a single valley for simulating this amplitude modulation. All right, so with this function, you can create these synthetic sounds. And once you have that, it's useful to put together, once you have the sounds you want to use in your experiment, the easiest part is, or easiest thing to do, is to put it into a single sound file. That then you will go out and play that sound file. instead of having a bunch of different sound files, each one for different examples of the sounds you want to evaluate. All right. So you can put all those sounds together with the function master sound file. So this taking the output of the previous function, and you also need to provide the duration of the gaps or the silence intervals between the sounds. And then the function will do this to concatenate all those sounds in a sequence, having those gaps in between. And in addition, it will add these markers that I call like acoustic markers, which is literally marker that says start and another one that says end. So start and end marker. And the function of these markers is going to be helping people to find the position of the re-recorded signals once you did the playback experiments, right? You go out, re-record the signals, and then you want to find the position of the signals, your test signals in those new recordings. You can do it, or it's gonna be easier with these markers. So it's gonna use cross correlation to do this, but I'm gonna show you, actually, maybe, no, it's not the next slide, but I'm gonna show you how you do that. But that's the function of those markers, right? And the other thing here is that it standardizes the amplitude. So all sounds are played with the same maximum amplitude, and the markers are played with a slightly higher amplitude. So it's more likely that you will record them at farther distances, even if the signals have been degraded, right? And they also have a low frequency that we know gets less degraded, and that also helps to make sure you will record those markers and you will be able to do this annotation regardless of the amount of degradation that you get on the signals. All right. So when you create this master sound file, it also returns kind of an annotation table or selection table, like in the Raven jargon, which basically tells you for that particular output sound file where is the location, what is the location of the different test sounds in time, right? We have these columns, start and end, and in frequency, bottom frequency and top frequency, right? Kilohertz. So basically it outputs a wave file and then also this table that tells you in this particular wave file, these sounds are located in this particular coordinates, right? And it also tells you kind of the treatments, right? The structure of the different sounds, like the duration 0.1, well, in this column, sound ID, sorry. The duration, the frequency, if they are tonal or modulated or amplitude modulated, all right? And it also includes the start marker and the end marker, and you will see why, all right? So when you do this master sound file, you get the sound file and also the annotations related to that sound file. And also important to clarify here, I'm using synthetic sounds, but you can do this with recordings, with your own recordings or something that you downloaded from somewhere else, right? It doesn't have to be synthetic sounds. It just simplifies like showcasing the package now, but of course you can do it with natural sounds as well. And it will take a similar workflow, right? You just, instead of synthesizing, then you bring your own annotation tables and sounds. All right, so after creating the master sound file, you need to go out, play the sounds with the speaker and re-record those sounds at different distances, hopefully several transects, and so on, some replication. But once you do that, you're gonna end up with a bunch of sound files with the re-recorded sounds, right? And then you go back to the lab, and the next step is trying to find the position of the sounds in those new sound files, re-recorded sound files, right? And that's not a trivial thing to do, and it can be very time consuming if you do it manually. And largely due to the fact that the signals you are expecting them to degrade, right? So sometimes you won't even be able to see them clearly in the spectrograms, which that's kind of the visualization we use to do these kind of annotations. So this can be tricky, and it's actually, I think, the part in which people get stuck most of the time, and can be very time consuming depending on how you do it. But the package offers some tools to simplify this process, which are related to those acoustic markers that are mentioned at the beginning. So first, I have this diagram down here. And what I'm showing you is in blue is the master sound file, let's say the spectrogram of the master sound file. And we have a bunch of signals that I call here ABC, and then the markers, or start and end marker, right? And below you have the one of the re-recorded sound files, in which you expect to see the same sounds, right? You don't know where, but you know that the time distance between the different sounds there should be the same that the one that you get in the master sound file, right? So the logic here is like if you find one of those sounds in the recorded sound file, then you will be able to find any other sound if that distance remain fixed, right? Which is the expectation. So that's the logic that we use for using these markers and um, based on the position of the markers, finding the position of any other sound in those re-recorded sound files, right? So the function find markers will take those master annotations that I showed you before and a bunch of test files, these re-recorded sound files, and it will look for both of these markers, their start and end, using cross correlation. So basically, taking both markers from the master sound file and looking for the position of those markers in the re-recorded files. And once you do that, there is another function. Well, the output of find markers is going to be basically the position of the markers. And with align test files, you can then figure out the position of any other sound from the master sound file, but now in the recorded sound files. Okay, so basically something like this that I'm trying to animate here is once you find one of the markers, then it's going to be easy to find the rest. So that's the logic here, and it tends to work pretty well. I will show you some data of even recordings at 100 meters. You can still see the marker. All right, and once you have that, then you're gonna have annotations for the position of all the recorded sounds, right? Which, as I said, that was the trickiest part, and I know, like in our software, that usually takes like depending on how much data you have, but can be days or weeks or I don't know. It can take a long time if you do it manually. I guess that's what I'm trying to say. But once you have that, then the rest is pretty straightforward, right? Because you have your test sound files, the recorded sound files, and you know the position of the sounds there, and then you can compare the structure of those recorded sounds to the original sounds, right? And actually, something that I didn't mention is that in these playback experiments, people tend to do a reference sound file is going to be recorded at 1 meter, right? So to account for variation that you have in the recording equipment and so on, so people tend to do that. But that's going to be your non-degraded example of the sounds, and you're going to compare that 1 meter with the rest of recordings, right? Re-recorded sounds of increasing distances. Okay. But then, as I said again, this is going to be straightforward once you align things, once you know where they are located in those sound files. Of course, in some cases, if you have lots of noise, background noise, or the degradation was too strong, maybe the alignment is not perfect. And alignment, I said, like this matching of the master sound file and all the recorded sound files. So there are functions that can allow you to double check this, right? You have just to trust things are right. For instance, this function plot aligned sound, it will take the output of the of aligned test files, what we got here, and then graph the first few seconds of the of each of the recorded sound files. Well, not the first few seconds, but the sometime around the marker that was detected, right? You can choose whether you want the start marker or the end marker, but it will plot the region where the marker was supposed to be located, and these lines with also the labels of the different like sound IDs that we have here in white. So basically, allowing you to double check if where the marker is supposed to be is actually there, right? Like in this case, we have this star marker in the right position with the right label. So that's one way in which you can double check things are right. Um, but in some cases, the detection is good, but not. It might need some adjustment, right? Um, and in small differences, small like changes, like an offset in the position of the signals, might have some impact in the measure of degradation that you're gonna do in downstream analysis. So you really want to make sure you are getting the correct position of the signals. So degradation is not measuring misalignments, but actually changes due to propagation in the habitat, right? So you can further like improve those positions with this interactive function that is called manual realign. And I'm showing you here. I don't know if you see this. This is an animation on how to use that function. The function will show you like these two panels. In top, you will see the master sound file that is being used to to figure out the location of the sound files in any other recorded sound file. And in the bottom panel, it will show you one of the recorded sound files at the time, and trying to show you the position that matches for the markers, so you can compare how it looks in the master sound file to the position or the sound that you got by aligning this automatically in the bottom panel. All right. So basically, you see in these two cases, we have the start marker and we have any other signal here, but there are some small misalignments um, that you can fix or you can adjust a bit with these options you have here, right? So you basically just click on this. You can stop at any time. You can move like big jumps in time to the left or right, or smaller jumps in time till you feel comfortable with the match in time between the positions in the master sound file and those in the recorded sound files. And once you do that for one file, you can move to the next one with this option here, next, and so on, till you go through all the recorded sound files and you make sure the alignment is the best you can based on the similarity of the positions between those sound files and the master sound file. So I found this really helpful, and even when you have a good alignment, automatic alignment, or finding of the position of the sound files, this is also a good thing to do just to make sure things are right, and make you feel, like that you can trust the data and is the best you can. You got the best data you can in terms of the position of the recorded sounds. So all right, so I've been showing you this first part of how to prepare the data and how to, once you've recorded, how to find the positions of the sounds. And then I'm going to show you more about the degradation of the sounds. Once you know where they are in the recorded sound files, then we can start exploring how much they degraded due to transmission in the habitat. So as I said at the beginning, I'm going to show this with this case study on the effects of habitat and acoustic structure on signal propagation, right? And for this, we created these synthetic sounds that vary in frequency. We use 20 different values between zero point 0.5 and 10 kilohertz. Um, we also have these sounds vary in duration with these two durations, 0.1 and 0.2 seconds. Um, the sounds also vary in frequency modulation, having pure tones and frequency modulated sounds. And finally, we have some examples of same sounds but with amplitude modulation, right? So we both have flat amplitude envelopes and modulated amplitude envelopes, just to so we can assess what is the effect of these different features in the propagation of the signals, in how much they get degraded, right? In the specific habitats. And so we, as you know, like this function that creates these synthetic sounds will do all possible combinations of features that you are simulating. So we ended up with 160 unique sounds, right? And we use this to explore the effect of the structure of the understory, doing transects, open and close understory. These playback experiments, playing and re-recorded the sound files at increasing distances, and we used four distances: 10 meters, 30, 65, and 100 meters. Actually, this was kind of the first experiment I did using the package. Now I usually don't record as far as 100 meters. With few distances, you can still get a good sense of how much the signals degrade. And we did this in a city forest. It was actually like a park in Mexico City, the Bosque de Tlalpan. And it has this serried shrubland, which is pretty interesting habitat with some parts that are very dense in the understory. So we did the playback experiments there. And this is the data that I'm going to be showing you. But still, while I'm showing you the tools, the functions that we have in the package. All right. So, well, this visualization actually is something that we included in the paper. There's no function that does this exactly, but I'm gonna show you some functions that allows you to do a similar comparison. But this I included here just so you can see have like an overview of the data. Which I think is pretty useful, and I think we should be doing more often, not only for this kind of experiments, but in bioacoustics in general. We are so lucky that this behavior can be so nicely represented in a spectrogram and have so much information about it. So we should do this more often. And in this case, I think is pretty informative. So we have these two columns, this should be understory, actually, open understory and closed understory. And then as you go down in the rows of spectrograms, distance increases from 10 meters to 100 meters, right? So basically, we can see and then in the spectrograms, we have the first 5 seconds of the recorded sounds, right? Or sound files, as you said. So we can see here the star marker, and we can see the sequence of test sounds after the marker, right? So if we just focus on this first column, we can see that as you go farther from the speaker, things get degraded, which makes sense, but not equally, right? Not like homogeneously in the frequency range or axis. Higher frequencies get more degraded than lower frequencies. And we know that's what we should expect, but I think it's pretty cool to see it and not having to trust theory, but you can see it with your own eyes. So that's pretty clear here. So the signals are being degraded as you move far from the speaker. And you see the same pattern here when close habitat. And I didn't explain this, I think, but basically to have this open and close understory, we did the playbacks on trails in which we didn't have any vegetation between the speaker and the mics. And for the closed understory, we did it off trails. So we have all those shrub in between. All right. So we can see the effect of that vegetation by comparing these two columns, right? And you see the degradation is way stronger when you have this vegetation in between, in close understory. Not only for higher frequencies, but even for lower frequencies. Um, and another cool thing that I see here is that you can get these markers even at far distances, right? Like 100 meters, you can still get a nice spectrogram for the markers, and we know that that's what is needed to find the markers, right? Because it's using cross correlation, it's using spectrographic cross correlation to find the markers. So if we can see them, it's likely that the automatic process of finding the markers we also do a good job. As I said, there are functions to do similar visualizations in the package. For instance, this function plot degradation is using these notations that we got after aligning the re-recorded sound files. But this one is taking one sound at the time, right? And in this case, we have the distances in the columns from 1 meter to 100 meters. And then we have a single example sound, as I said, 8.5 kilohertz sound, and we know that is modulated in frequency. But the first row is for the open understory, and the second row of spectrograms for the closed understory, right? So we can see again, well, the amount of degradation that this sound is suffering as it moves away from the source, but also that this degradation is stronger in close understory, as we expected, right? Like this is not something surprising in that sense, but I think still pretty cool that you can actually see it. And it gets actually very interesting if you compare, for instance, sounds with different frequencies. So the top two rows of spectrograms are the same that I just showed you, an 8.5 kilohertz frequency modulated sound, in which you get more degradation in the close understory. But the bottom two lines of the spectrograms are showing you the same, but for a 3 kilohertz sound. So a similar sound, but which is frequency modulated, but with lower frequency. And you see that the degradation is not that strong on either the open understory or closed understory transects. All right. So again, showing you, but pretty clearly, the effect of that vegetation in the propagation of the sounds. There are visualizations you can do. For instance, this function plot blur ratio will create these image files for each of the test sound files. Oh, sorry, for each of the test sounds, in which you can compare the amount of degradation by looking at the amplitude envelopes. So I'm going to explain you, I'm going to walk you through the image. Here we have the spectrograms of the reference sound, and remember reference sound refers to the recorded sounds at 1 meter, right? So where there was no actual degradation due to the habitat. And you just do that to account for equipment effects. So you have that reference sound, and then you plot any other the same sound that increases distances. In this case, we are comparing that reference sounds against the one recorded at 30 meters. And you can see the amount of degradation that they suffered. But this measurement, blur ratio, is actually related to the modification in the amplitude envelope, right? And so you can see two amplitude envelopes here together. In purple is the one from the reference sound. So everything here in purple is related to the reference sound. So this is the amplitude envelope of the non-degraded signal. And then the amplitude envelope of the same signal after traveling 30 meters in that habitat. And the mismatch between the two is what is used to measure this blur ratio. So it's basically the area of the mismatch divided by the total area of the envelopes. So that's what is being measured with blur ratio. But you can actually plot this and have a sense of how much degradation is happening and which parts of the signals are being affected and so on. And of course, the package also offers functions to measure degradation. Right? I won't go through what each of these different functions is measuring in terms of degradation, but I think as a general idea, they try to quantify how much the intensity of the signals was affected. For instance, with signal to noise ratio. Blur ratio is a bit more specific, not only how much it was affected, but how homogeneous was the effect of the decrease in intensity on the signals. And there are things related to the distortion of the signals. For instance, cross correlation. This is just a regular spectrographic cross correlation, or tail to signal ratio, which will look at the tail of the reverberations and measuring the relative energy in those reverberations compared to the actual signal, right? So measuring distortion, reverberation, and decreasing in overall intensity. That's what is being measured by these measurements. And they all work on the same objects, right? And whatever you have, you use to save the aligned sounds after finding the markers. That's what you use here. And this is only using, for simplification, I'm running all of them at once using a pipeline in R. Right? But that's how you can measure this degradation. So we did that with our data from Talpan, looking at the effect of the understory structure, the closed versus open understory. And these are the results we got for that. So there is a lot of things going on here, and it's gonna take a while to show you this, but it's important we all understand what is being shown. So we wanted to understand, as I said at the beginning, the effect of the habitat structure, closed or open understory, and also the structure of the signals related to the frequency, the duration, the amplitude modulation, and the frequency modulation into the amount of degradation that they suffer, right? And that's what I'm trying to show you here with this plot. So what I'm showing you here in these cells is the effect size, which tells you the magnitude of the effect of the predictors, which are, as I said, habitat or the acoustic features, into the amount of degradation. Okay, so we have the predictors here in the x-axis, and in the y-axis we have these different ways in which we can quantify degradation. So I use some raw measurements of degradation, as I already talked about blur ratio, but there's a couple more. Tail to signal ratio, I already explained it, and excess attenuation, which measures how much extra attenuation you got in top of the one that is expected just by simple spherical spreading. So we have these three measurements, and I also created like an overall degradation metric by taking all the degradation measures that I showed before with the functions and summarizing that into a single vector using principal component analysis. And if you are not familiar with that, I can explain how that works later. But basically, doing some dimensionality reduction, so you can take all those different features representing degradation and put it, condense it into a single vector that you can use in a regression model, right? So these are all regression models. And again, the cells gives you the answer. What is the effect of those particular predictors on those particular measurements of degradation, right? And I use a color grading just so it's more friendly, so you don't have to interpret the numbers. But the greener, the stronger the effect, positive effect, and if it gets a bit purple, is you have a negative effect. And the last piece of information to understand this graph is the font color. So the ones that have a gray color means that the association was not significant, right? And the other ones you have a significant association. Okay, so once we understand what is being shown in this graph. We can see that the most important predators to explain the degradation in the signals were the habitat structure. You see, like the mostly green for across the different measurements of degradation, and the frequency, right? So most of the degradation that we quantify here comes from the whether we are in an open understory or a closed understory. And of course, closed understory produce more degradation than open understory. And the presence of that vegetation when the sound is propagating significantly affects, and apparently more than pretty much any other feature in the signals. Because these other predictors here, frequency, duration, amplitude modulation, and frequency modulation, are not features of the environment, but features of the signals themselves, right? And from those features of the signals, frequency, not surprisingly, is the one that explains most of the variation or the degradation that the signals suffer, right? So a good amount of the degradation comes from the variation in frequency that we have in the signals, and more specifically, higher frequencies suffer more degradation than lower frequencies, something that we already knew as well, right? But it's good that we can see that the package is able, and the measurements that are included in the package, as well as the process of finding those sounds and creating all the data for being able to test this, seems to give you a reasonable answer, right? And then you have other like results here that are, I don't know, not that expected but interesting, like amplitude modulation. So it seems to when you have modulated signals, seems to help a little bit with the blurring in amplitude. And frequency modulation has the same effect. Sorry, amplitude modulation and frequency modulation have increased the degradation when it's measured with blur ratio or overall degradation. That's what we have with these four things here, which was expected. What was unexpected was these results down here. So negative effect sizes means that you have less degradation in this case when you have amplitude modulation or when you have frequency modulation, but mostly in terms of the amount of attenuation that you have in the signals. All right. And I can show you, I can share with you the paper in which we describe these results. So there's a lot here going on. But in general, the quantification of degradation matches our theoretical expectations in a pretty good manner. Um, and so the overall process of generating these sounds and running these experiments getting this data from those recorded samples seems to work in a good way. All right, so I'm gonna keep it there for in terms of result. There are many things that can be shown with that data, and again, I can point you out to the paper. But in terms of the performance of the package compared to other computational tools for doing similar analysis, so we compare baRulho with what actually the only one that we knew about, the only software to do similar analysis, which was SIGPRO. And we took like a subsample, I think like 50 sounds that we analyzed manually with SIGPRO, and it took us 861 minutes. Actually, this was my student, like Marcos. Thank you, Marco, for doing this. He learned how to use the package, and then he ran the same the analysis on the package, and I mean on SIGPRO, and then on the baRulho package. And the same data set with baRulho took him like 7 minutes. So it's like 120 times faster. Of course, you need to get familiar with all pieces of software, but I think that's realistic that it can go way faster with baRulho. And mostly because the annotation and the measurements have to be done manually with SIGPRO. But baRulho offers a way in which you can do it in an automatic way, just by making sure you find the position of the signals, right? So that's why I think it's an important contribution of the package, like simplifying these things and making the analysis more friendly. And in terms of the similarity between the measurements you get with the two software packages, um, we got a pretty good match between the measurements, for instance, the tail to signal ratio, a correlation of zero point 0.8, signal to noise ratio, 0.94, blur ratio in zero point 0.8, and excess attenuation close to a perfect correlation. So basically, the two software packages are measuring the same things on the signals. Right? So overall, like it gives confidence that it's a sound approach to measure degradation on acoustic signals, the package baRulho. Okay, so I think we can say that the quantification seems right, right, matches our expectations, and it also um, is similar to what we get with other software doing similar analysis. The performance seems to be improved by applying these methods of automatically finding the signals in the recorded sound files, which dramatically improves this. I would never do this if I wouldn't be able to do it like automatically, because it can be pretty time consuming. Like can be, I've seen people doing it for months or even years. I don't know. It takes a long time. Can take a long time if you do it manually. And overall, I think is it helps to expand the bioacoustician toolkit by allowing to do these experiments in a more friendly way. It also adds these new measurements I didn't mention, but only these four measurements are included in SIGPRO. So the rest of the measurements you do in baRulho come from other implementations of measurements of degradation, plus some new additions that people didn't use before. So it comes with these new measurements that allows you to look at different dimensions of this degradation space. And of course, visualizations that allows you to double check and understand what is going on with the signals when they travel and degrade in a habitat. Okay, and the last thing that I wanted to show you is some usage of the package in my own research. And this is related to some work we are doing on the cultural evolution of songs in Hamburg. So this is the work of the for the thesis of my student Marco Quiroz, which has done a great job on this. It's been pretty tricky. And he did all these playing by experiments, and now we have the data, and we have found some very interesting results that I'm going to show you here. So these species legs, so it displays in these core ship assemblies in the forest. And what is interesting, as these guys, the long-billed hermit, as many other hummingbirds, they learn the song. They socially learn the song, so they kind of copy the song from other individuals in the leg, right? But there is a lot of variation. Like within the leg of 10, 12 individuals, you can have several song types at the same time. Like some, you can call it like song-type neighborhoods, like subgroups of the leg singing different variants of the song, right? So that's what I'm trying to show you here, like the crazy amount of variation that you can see in this species, right? So I think there are like 100 and something different song types that we have recorded in during several years of fieldwork before Marcos, but we still keep doing this. So there is so much variation in the structure of these signals across space, within legs, between populations, and also across time. When they a few years pass, the structure of the signals will be modified. Some sometimes will disappear. You will have some new contacts and so on. So there is so much variation. And we wanted to understand like where this high diversity comes from, and some aspects of this interesting process that we can call like a cultural evolution process of creating these sound types, which I think is better represented with this graph. Here in the horizontal vertical axis, we have the different sound types, and the bar means the years in which each of those sound types was found at the leg. This is for a single leg. I'm not showing you the entire data. For the entire data, we have like 12 different legs. But for one of these legs, we can see that most sound types, they just last for 1 or 2 years. But there are a few sometimes that can make it for longer. They can persist um, many more years. In some cases, we've seen like even 10 years. So there is an important amount of variation in the time that they can survive, right? Like the lifespan of the sometimes. Again, when we're talking about the sometimes, these cultural traits, but not the individuals themselves, right? So how long they persist in the legs is quite variable. And you can actually split this into, well, if you want a binary representation of this persistence, that's what we did. Have some sometimes that we can call like low persistence, like the 1 or 2 years sometimes. And then what we did is classifying the ones that survive or persisted for more than 5 years as high persistence, right? So we have these two groups in which we wanted to understand what was why they survive longer, or why we have this variation in the persistence of the sometimes. So we related this thing to degradation. And the mechanism that we think or we propose to link these two things is that signals that get more easily degraded are less likely to be copied by other individuals. And again, will be less likely to be to persist through time if they are not copied, right? So that was the mechanism that we expected to link these two things. And here I'm showing you just one result from this study. Um, we have in the x-axis the different sum types that we used. We have like 20 sum types for high prevalence. Well, we should call it persistence here, high persistence, and 20 sum types of this low persistence category. And then we measure degradation in the same way that I explained before, doing display experiments, we're recording them, measure the degradation features, and then we summarize degradation in a single principal component, right, in single dimension. And that's what I'm showing you here in the y-axis, right? Um, and as you can see, the high persistence are in blue and low persistence in sky blue. So there is no clear pattern as we predicted that the ones with higher persistence will suffer less degradation, right? So they are all equally likely to suffer degradation. So degradation doesn't seem to explain why some types are more persist for longer than others, right? So it's not explaining that part of the cultural evolution process, which is still an interesting result because we know that in evolutionary uh, the process of creating variation in acoustic signals, degradation seems to matter in well, often, not all cases, but we often see an association between the propagation properties of signals and how likely are they to occur in specific groups living in specific habitats. But that's not the case for this cultural evolution as far as we know. But what is for me is also pretty interesting is that the amount of degradation is affected by this cultural evolution process. So these lines here on the top are showing you, are linking groups of subtypes that significantly differ in degradation, right? So for instance, the one with the lowest degradation significantly differ from this one here. And so basically groups of some files at the two extremes of the range of degradation that we observe seem to vary significantly in the amount of degradation, which we take as indicating that the variation that is generated to cultural evolution is actually affecting the propagation properties of the signal, right? So it's not like a neutral process that is not doing much with the signals, but is affecting a very important feature of them, like how easily they travel to the habitat, which in turn is related to this idea of the sounds being adapted to specific acoustic habitats where they are found. So it seems that there are other processes that can play a role into how that happens, right? And not only this acoustic adaptation of signals to the environment is shaping the propagation properties of signals, but other processes like cultural evolution are also affecting that. And that's what we have so far. Well, Marco is going to be defending his thesis soon, and I can share, well, yeah, the link with you guys if you want to look at that. It's going to be very interesting. Um, yes, and that's what I have for the research. And well, thank you guys for staying to the end. I just want to acknowledge like several groups of people, institutions that help us with that. Like I work at the Centro of Neuroscience Research Center here at the University of Costa Rica, um, and also the Universidad Nacional Autónoma de Mexico. So we did a workshop there, and that's where we did all these playback experiments, and also we're able to work on the package a bit more. And also our packages that we always use when we do acoustic analysis, like C-Wave and Tuner, they're always like incredibly helpful for doing these kind of things. Well, that's it. Yeah, I'm happy to take any questions.
Thanks so much, Marcelo. We've got time for a few quick questions here. You see the emoji claps popping up. Um, Thanks everybody for sticking around. Good presentation. I'll start with this question from Kelly. Um, in your opinion, should these degradation measurements be taken at different points during the deployment of your ARUs? How would you handle the scenario that they have where they deploy and there isn't much grass, but then when the detectors are picked up, the grass has grown much higher than when it was deployed? So situations with extreme vegetation change.
Oh. But you mean in terms of measuring degradation or related to what aspect of research?
Yeah,
Um,
Kelly, feel
I know
free.
you are reading that, but
Yeah, feel free to pipe up, Kelly, if you want to expand. Um, they ask, should these degradation measurements be taken at different points during the deployment?
oh yes, different points in your planning. So basically, people tend to record. You have like a transect in which you will do the experiment, and then you have mics at increasing distances from the speaker. And I think it's common to have two, three different distances. And my experience is that with two or three distances, you will get a good sense of what is going on. And usually, I don't know, like 20 and 40 meters. People like to have like more rounded numbers. It doesn't have to be that way. But with some, if you know that signals will be degraded at some point, then if you record across that range a couple or three different points, then it's likely that you will pick up the statistical signal of the degradation and the factors that are affecting the degradation. So you don't have to go all the way to a point in which you barely hear anything or detect anything.
Yeah.
Um, yeah, I guess.
And in this case, Kelly, it's talking about location and time. So in a
We'll see.
deploying in a place where uh, the grass grows a ton over the length of the deployment. And so the vegetation really changes at different times in the year.
I see. Yeah, it sounds to me a bit like a coming from passive acoustic monitoring because length of the deployment,
Oh,
we
right, right, right.
do this like in one day, like one morning, one afternoon, and
Yeah.
so there's no time for things to grow.
Yep.
But I think it's still like a very valid question because, yeah, the habitats are not stable. Well, depending on what you do, I think you might want to look at the degradation, the effects of habitat in its different states, right? Like, for instance, I know like something common here in the tropics in like dry forests that they drop the leaves and that dramatically changes the structure of the forest and then it's pretty interesting question how much that affects the entire process of adaptations of the acoustic signals of animals to that. And in that case, it would be a good idea to do the experiments on both forest with and without the leaves and see what happens. But yeah, I guess it's something that you also want to take into account. And if for doing that, you will need to redo the experiments on different variations of the habitat.
Yeah. Yeah. Similarly, Rachel asks, I'm doing an experiment where I'm playing bird calls from different distances at different times of the year. Uh, does the package compare between time points? Would you just do them separately?
Timelines. Um, no, you, well, that's more like a statistical trick. You just, well, it will, the package will do all these steps that I show you from finding the markers. Well, of course, producing after doing the the experiments, finding the markers, aligning the sounds, creating a single annotation table, and then on that measure degradation. It will do it on any recorded file. So it could be a recorded file from single transect for 100 transects, from a single point in time, or from many years. It doesn't really matter that much, or at all, as you said. It will do the same process, the same yeah, measuring of those features on all the sound files that you provide that were the result of these playback experiments. So it could be many years of data, it could be like a single day thing and many transits. But to account for these things, then that's more related to how you manipulate the data and put that into a statistical model. You need to ask the model, I want to include I don't know, like the season as a predictor, for instance. But that's not that much about the package. It's more about
Right.
handling and using the data after you got it from this package.
Dimitri asks, how does this method compare to propagation modeling?
To what?
Propagate to propagation modeling?
Propagation. Huh. Um, that's a good question. I don't know much, but and I would like to learn. But I think so. There is no modeling going on. I guess the only thing that uses some like theoretical expectation is the excess attenuation. So you have you have this like a regression, mathematical model that tells you how much attenuation you will have for a frequency of this after traveling this distance, and then you compare the observed attenuation to that theoretical attenuation, and that's it. But for the rest, there is not much modeling going on. It would be interesting, and I think this is in part to the complexity of the environments and how hard it is to model what is going on, interaction of those sounds in these very complex 3D spaces in natural habitats. Yeah, that's what I can tell you.
Matt and Tim have uh, similar-ish questions about can you use if you're are you able to use pre-recorded and isolated vocalizations rather than ones created in the program? And if so, does it allow you to add your own markers, stipulate your own markers in your recorded sound files?
Yeah, that's a great question. Yes, you can use your own data, acoustic data for that, and you just do it in the same way. Once you have those sounds annotated, you need in R like a data frame or a table in which you have the names of the sound files and the positions of the sounds you are interested um, in and time, and if you want in frequency. And then with that, you can tell the function like make or the master sound file to create a single sound file containing all those things plus the markers, right? And it can come from several different sound files. It doesn't have to come from a single one. And the documentation of the function can guide you into doing that. And you don't need to use those markers. You can use your own. And sometimes I've seen like some issues like people forget to press like a rec on the right time. So we skip the first marker and things like that. So you can tell the function, I'm gonna use the sound number 3 as the marker to align things. Or you can create your own. The good thing about these markers is that they were created by just taking the images of the words and doing an inverse Fourier transform. So converting that into sound, but it's such a weird sound. It sounds like a robot. And
Mhm.
then there is nothing, it's very unlikely that something in the environment will look alike, right? So it's hard for the cross correlation to miss something else, to be confused, you know. But you can use your own markers. And I've been thinking about adding a third marker. The other thing that the package um, does, and which is a good idea using the markers that the function provides, is that it also measures the distance between the start and end marker and compares the distance in time, right? And compares that distance between the original master sound file and any re-recorded sound file. And the distance, if the detection was correct, then the distance between those two markers should be very close to the one in the master sound file. So that comparison gives you also a good estimate without even having to look at the spectrograms, whether the alignment was good. So that's another advantage, but it was too much to show here. But that also helps you to get to be confident in the aligning of the annotation of the recorded sound files. But you can use your own demonstration.
Jay asks, how does the synth sound function distribute sound energy across harmonics when they're included?
There is an option to include harmonics that it didn't show. And for me, I think the harmonics like a simulation is very rudimentary, it's very simple. But it just I don't remember how I did it, but it's just like a fraction of the amount of energy in the fundamental frequency. And as you go up, it will decrease. But I don't remember the exact like formula that I use for that. But it's not a fancy implementation of that, but you can still have some harmonics. You can decide, I think, um, which harmonics you want to include in terms of, as multiples of the frequency of the fundamental one. But what is tricky for me about harmonics is to the fact that they are always like higher frequencies and higher frequencies degrade more. So it's a tricky thing to compare between things that have and not have harmonics. And so that's why I did include that here. But at least the function includes something to do that, but it's not very fancy. But I think it can help working with harmonics. Um, yeah, no. I don't know if I answered your question, but yeah.
And then maybe one more. Somebody's interested in the spectrogram matrix you made. Um, they're working on differentiating the song repertoire between two birds, comparing the inter and intra-specific song variations. What analysis did you do for that intra-specific song variation?
Huh. Um, it depends. But so the thing is that we have in like the bioacousticians toolkit, we have several different tools to measure structure, right? But they each one look at different aspects of the sounds, right? It could be the overall spectrogram, but it could be also just looking at specific things on the signal, like peak frequency, duration, distribution of energy in time and frequency, and so on. Or it could be variation in the frequency contour. You can use like dynamic time warping or time series analysis to do that. But they look at specific features and then they give you, in most cases, like a good measurement of the similarity or the overall structure in those dimensions, right? What is tricky is that you need to use a tool that is going to measure the features that are relevant for the variation in your system, right? Because you can measure something else and it will tell you there is nothing happening here, but you are not looking at the right at the right feature. It's like, like you're comparing two subspecies of like lizards and they vary in the length of the tail, but you measure the head. It sounds like a trigger, right? But it's more cumbersome with the acoustic signals that it's not that easy to look at those different dimensions and figure out which one is the most varying feature. So that's the previous question you need to answer in order to think about which is the method that is going to tell you that. But that said, like people have used these measurements, like the ones you get with Raven, like multiple measurements on the distribution of energy in time and frequency, and the frequency contours and things like that to come up with like a multidimensional representation of the structure of the signals. And it seems to be work decently in many cases, right? So depending how you do it, that's a way to do it, but doesn't necessarily mean you will hit the right spot, the sweet spot there. But that's kind of a default approach that people in bioacoustics have been doing. What I
Yeah,
would suggest is like trying to graph somehow or compare the similarity that you get with the specific measuring tool to the ones that you come up with your head by just looking at the spectrogram, right? So a good measuring approach will be something that tells you two things that look different are very different according to that approach, and two things that look similar, they are similar according to that approach. Um, so that's a straightforward way to ground truth what you are doing, to make sure you are measuring what you want, and the numbers are giving you something similar to what you see in the spectrograms.
cool. They also said they're realizing song differences might be affected by the habitat, so they're excited to try out the package.
Oh, okay, cool. Yeah, yeah, that's where this package comes like handy because then you can go play those sounds there and see you have a clear expectation, right? Like in those, sometimes we'll do a better job at propagating a certain habitat.
Excellent. Let's call it there. Thank you, everybody, for attending. Thanks again, Marcelo. We'll get this uploaded to our YouTube. Um, hopefully, see many of you at the next BioAcousTalks. I believe it's the 18th, back at 4 p.m. Eastern. And then we're going to reschedule the Raven team to talk about Compass and Expedition soon, hopefully. So awesome. Well, thank you so much again.
All right, thank you, Karen.